Welcome to the Granatum tutorial. We will start by going to the website. We have provided some example data directly with the site, either by clicking the expression table link here or the metadata link here, or you could get these in batches 1, 2, 3, 1, 2, 3. I'll explain this in just a moment. So let's go ahead and just download the expression table and the metadata table. And since this is human data, we're going to use the human, not the mouse, the human species. And next we'll select the expression table that we just downloaded here. Browse, document downloads. We have our expression. Wait for that to load until it says upload complete. Now the metadata. And when we're ready, now that we're ready, we can do add dataset. And we will see that we've added one dataset, total number of genes, total number of samples, number of cells that were sequenced. And here we're seeing a preview table of what's inside the expression table that we uploaded and what's inside the metadata table that we uploaded. So this is the cell identifier. And for example, this is the type. It's PDX primary cancer or PDX metastatic cancer. And here in our expression table, going back, here again we have the IDs on the top. And here are the genes. And these are the number of reads mapped to the genes per cell. Now, we just uploaded the entire 118 samples. However, we can also do this in 3 batches. And so let's go back, we'll reset. Now we're going to select the 3 batches of the expression table. 1, 2, 3. And 3 batches for the metadata. 1, 2, 3. And these, when they're done, we can go ahead and start by selecting the first expression dataset and its complementary metadata, number 1. And we'll go ahead and add this dataset. Now here you see we have 36 samples instead of 118 because we're just doing we've just uploaded the first batch. Next we'll upload the second batch. So to do that, we go down to add another dataset. And brings us back here where we can browse to our second. Here. And our second. We'll add dataset. Now we've added this second dataset with 47 samples. And to complete it, we're going to add a third dataset. So expression table number 3 and metadata table number 3. Add dataset. And so we have the third dataset now uploaded with 35 samples. And we're going to see the expression table and metadata table of the last dataset that was uploaded. So in this case, it was dataset 3. And these are actually all the patient metastatic type cells. So now that we've uploaded all 3 batches and we have our 118 cells, we're going to continue to the next step, which is the batch effect removal. So in this step, what we're seeing here are expression statistics for a random sampling of up to 96 cells to make it legible, to keep it legible. If we'd like to resample, we can resample and get another 96. What you may notice here is that there are actually a couple of cells here which have pretty poor or low levels of sequence expression, and of expression, that is. And this will show up in the next outlier removal step, actually. But for now, we're going to go ahead and do batch effect mitigation. And when we've uploaded 3 separate data sets, we get a batch factor, which is labeled dataset. So our 3 files essentially are showing up as 3 different colors here. And we can use this, or we can use any other factor in the metadata. So let's just go ahead and use the dataset factor and perform batch effect removal. And you'll notice that this batch here moves slightly. All right, now that we've done that, we can proceed. So go ahead and submit, submitting our data to the outlier removal step. So what we're seeing here are each of the cells and their reduced representation plots. And there are a couple of cells here which have probably had some problem in sequencing, and or perhaps capture, or maybe they died during capture. And we don't need these. We would actually rather remove these. So in order to remove these, we'll go ahead and select them. And actually we can select cells from either plot. I'll update. And we can deselect. Let's go ahead and select these. And then choose Remove Selected. This will replot the graphs. And now you don't get those 2 outliers, and those will be removed in the subsequent steps. So let's go ahead to the normalization step. Again, you're seeing the same kind of box plot representing expression levels per cell. Up to 96 cells are being shown, and you can resample from your data set of what was 118 is now 116 individual cells. And the orange dots here represent the geometric means. So for this example, we'll go ahead and just rescale all of our cells to have the same geometric mean. And you'll be able to see all the dots line up. If you want to do any of the other normalization methods, just click Reset to go back to the data that you came in with, and then you can click whichever normalization method you'd prefer. So for now, though, we'll go ahead and just use rescale to geometric mean and move on to the next step. So in this next step, which is going to be gene filtering, we'll be seeing a degree of expression basically on the along the x-axis, and dispersion along the y, or actually above the dispersion line here. So what we're looking at here is to remove those very lowly expressed genes overall that may not provide much information, and also those that are not very highly dispersed. That is, they don't have a lot of variation between the cells, and so they're not likely to be as informative in subsequent steps. So we can keep these settings here. And so we're starting out with 19,924 genes, And after this filtering step, we're going to be taking all the genes that are above this line and at this expression level and higher. And this will be 3788 genes that we'll take to that next step. So now we're at the clustering step, and we can choose what sort of labels we put on our cells here. For example, this is you could change it to just show which are metastatic and which are primary cancer cells, or use the 3 different categories. So we'll just keep with that. And then we'll choose a clustering method. So perhaps we can kind of see that there are 3 clusters in the t-SNE plot. And we'll go ahead and do a k-means on 3 clusters and run clustering. And we'll see that we pick up the 1, 2, and 3 clusters pretty well. The numbers indicate how the clustering algorithm has assigned each cell. So group 1, group 2, group 3. And these labels are from the metadata that was initially input. In this next step, what we'll be doing is we'll be comparing the 3 clusters for differentially expressed genes. Now we provide an option to change the number of cores, because the number of processor cores, because this step is computationally intensive, and so the more cores you can use, the faster it will process your data. But the more cores you use, the more RAM memory you will consume. So we'll keep it at 2 for our VirtualBox instance with about 8 gigabytes of RAM, and give it about 30 minutes to process all pairs between the 3 different clusters. So we'll go ahead and start that analysis and come back in 30 minutes. And below that are our differentially expressed genes. The tabs indicate which groups are being compared. In this case, 1 versus 2 would be the PDX meta down here versus the patient meta over here. And what we're sorting on is the z-score, and a positive value means that this gene is overexpressed in the first group. And if we want to see what sort of gene this is, we can click here and we get a link explaining to gene cards, giving some detail about the gene. And we can look at the others as well by just clicking on the other tabs. And below the differentially expressed genes table, we can see 2 buttons now, KEGG enrichment and gene ontology enrichment. This will look for the enrichment of differentially expressed genes in KEGG and gene ontology databases. So we can go ahead and click here and allow it to calculate. And we'll get a plot down here where the highest here is the most most enriched pathway, or we'll do gene ontology terms. And this is all relative to whichever tab you are using. So this is still group 1 versus group 2. And sometimes it can take a moment. And so here we have our gene ontology terms for which differentially expressed genes are more or less enriched. And at the bottom, we also have a download button. You can download the set of differentially expressed genes, the table, CSV table. So now we can go to the next step. So in this next step, we'll take all the differentially expressed genes and we can visualize the protein interaction networks between those differentially expressed genes. So if we use our mouse wheel, we can zoom in and we can select the protein to highlight it and the connections to other proteins, and the colors give an indication of whether the z-scar is high or low, whether it's up or down regulated in the described group, and we can click between our different groups, get different networks, and take a look. We can also, if the networks are very dense, we can move the points around to make them more visible. And once we're done, we can sort of move around. And once we're done here, we can proceed to the next step, which is the pseudo-time pathway construction. So in the pseudo-time construction section here, we're using all of the genes from our filtered selections. And what we'll see is a construction of a pseudo-time ordering the cells depending on their expression profiles. This can be used to trace a path of differentiation. For example, here we have our three different groups, and some of these cells may be closer to other groups around this branch point here. And as before, we can change how we label the points. And this concludes the walkthrough tutorial of Granatum.